In the following slides, I will explain the differences between the protein translation of prokaryotes and eukaryotes. To begin with, the Shine-Dalgarno is a ribosomal binding site that is only found in the messenger RNA or mRNA of prokaryotes. It helps start initiation of protein translation, as this is where the 30S ribosomal subunit aligns itself. The Shine-Dalgarno is usually located just upstream of the AUG initiator codon. If we compare this with eukaryotes have a 5' cap at the start of the mRNA. This is a 7-methyl guanosine structure, which is important for many reasons. These include translation initiation, as the 40S ribosomal subunit binds to this cap. It also acts as a protection for the 5' end of the mRNA. Eukaryotes also possess a 3' poly A tail, which prokaryotes do not have. This is added during transcription and is made up of a series of adenine nucleotides. These add stability to the mRNA, aid in translation termination, and act as protection for the mRNA like the 5' cap. The length of a poly A tail is usually approximately 200 bases, but this length decreases with age. As I said, the 30S ribosomal subunit is the first to attach to the mRNA. This is then joined by the 50S larger subunit. These work together and form a 70S ribosome, which moves along the mRNA to synthesize proteins. These are made up of both ribosomal RNA, or rRNA, and proteins which make up the 70S. Like in prokaryotes, the smaller ribosomal subunit attaches to the mRNA first, but in eukaryotes, this is a 40S subunit. It is then joined by a 60S larger subunit. They move together along the mRNA as an 80S unit. The final major difference is the place where the protein synthesis actually takes place. In prokaryotes, transcription and translation both occur simultaneously and in the cytoplasm. However, in eukaryotes, transcription takes place in the nucleus and translation in the cytoplasm.
Protein translation consists of 3 stages: initiation, elongation, and termination. The first stage of translation is initiation. During this stage, small and large subunits of ribosome join together with mRNA and tRNA to form the initiation complex. Firstly, the small subunit of ribosome binds to the mRNA strand, which is made up of codons. The small ribosomal subunit begins to move along the strand in 5' to 3' direction, stopping when it reaches the start codon. tRNA, which contains an anticodon complementing the mRNA initiation codon, binds at this point. The corresponding amino acid is attached to the end of the tRNA, which in this case is methionine. The large ribosomal subunit then joins the small subunit of ribosome. There are 3 tRNA binding sites within the large subunit of ribosome: the acceptor site, the peptidyl site, and the exit site, known as the A site, P site, and E site respectively.